Nice to see you guys. I know it's a very cold day. I'm glad that you're here. It makes the lecture much better for me. I hope bless you. I hope the utility of coming in this cold day is going to be positive for you. And I hope you had a good spring break. So I will be describing for you the motor system, a little bit more about the neurophysiology of the motor system and how it controls movements in the next 3-4 lectures. And I'll put together a homework for this week and next week as well. This week's homework is going to be on some of the material that I taught you a couple weeks ago. And next week's homework will be on some of the material that you'll see this week. So today what I'm going to do is describe for you the organization of the motor system. And so here we have our cerebral cortex. This is the cerebellum down here. This is the brainstem and the spinal cord. And I want to think about the act of making a movement like a reaching movement. So as I want to reach, what happens is that first I have visual information about the thing that I want to reach toward. And that visual information is information on my retina, which gets represented on my parietal cortex back here in terms of where information is, where this is with respect to my fovea. It's a representation of space. It tells me the thing that I want to reach toward is at this location. And you also see representation of value at this location. So when we talked about utility, we talked about stimuli having a value associated with them. So that directs my gaze toward them. Similarly, if I want to reach toward something, the item that I want to reach toward has a utility. And you see that neurons in the parietal cortex, they respond to locations of a stimuli if that stimulus has value because I want to reach toward it. So you have this visual representation of the world in your parietal cortex. In the motor cortex here, this is the central sulcus here, and the motor cortex is right in front of that, to the front of the central sulcus. You have neurons that go to your spinal cord and transform the information about where the object is to the action that I need to do, move my arm toward it. The information about how to activate the muscles, which muscle to turn on, which muscle to turn off to make that movement, you see it in the primary motor cortex. Now, there are these two other structures that are highlighted here. One of them is underneath the cortex here. You can't see it. It's called the basal ganglia, which we saw a little bit of information about it last time. Dopamine is there. That's the region that seems to assign value to the stimulus that I'm interested in. So the idea that I can describe utility of objects and the importance of those objects seems to be associated with the basal ganglia. This is the place also that affects an important disease called Parkinson's disease, where neurons die that generate dopamine. Another structure that's important for control of movements is the cerebellum down here. And this structure is not important for making a movement, it's important for making the movement well. So if you have a problem with your cerebellum, you can still make a movement, but you might have tremor at the end of your movement. You might not reach in the right way, so you might reach in the wrong direction. Some of the information that you need to do in order to compensate for the complexity of the world. So for example, say you pick up an object that's heavy. Well, that heavy object changes the dynamics of your arm. The fact that you can move with that heavy object just as well as with a light object is because your cerebellum tells you how to do it. We think that the cerebellum is important for learning how to do movements, like learning how to ride a bicycle, learning to ski, learning to play ping pong. Any sport that you would play with, you are stressing this region, the cerebellum, which is very central for learning to control. In fact, there are more neurons in the cerebellum than the rest of the entire brain. So more than half the neurons in your brain is here in the cerebellum. So we think this is important for helping you learn to control movements, coordination, and eventually the information from the motor cortex comes back down to the spinal cord. Before it gets there, it acts on regions in the brainstem here. And an example of that is that if I were to ask you to pick up a book, so you would have to activate, say, your biceps to pick up that book. Now, for you to do that, you would think that the motor cortex would just activate the arm. But it turns out that before your arm lifts the book, your leg gets activated. And the reason for that is because you have to stabilize your posture before you can generate forces. And that act of stabilizing your posture is done through the brainstem centers. Eventually, the information reaches the spinal cord, and then it gets to the motor neurons, and then it gets to the skeletal muscles. And so we're going to cover in my lectures the brainstem, we're going to cover the parietal cortex, we're going to cover the motor cortex, and we're going to cover the spinal cord. And then you're going to hear from others regarding the basal ganglia and the cerebellum. All right, so that's the overview of the next 4 lectures. And before we start, do you have any questions that I can answer for you? All right, fair enough. So we're going to talk about the spinal cord today. I don't know if this is, maybe, I don't know if these lights can be reduced a little bit so you can see this better. But regardless, here's a picture of your spinal cord and how it sends nerves that will act on the muscle. So here's the structure that holds up your body. There's the vertebra here that you see here. And between the vertebra is the spinal cord, that's perfect. This 1 centimeter diameter sheet of nerves that houses both the motor neurons and the sensory neurons, as well as inner neurons that are important for control of movements. So, you know, when people have back problems, it's discs that are separating the vertebrae that may give them issues with back pain and so forth. So the nerves leave the spinal cord. So here's a nerve that's leaving the spinal cord, and then they go out. So for example, here's the cervical spinal cord. Here's a nerve that's leaving the spinal cord, and it goes out all the way down to the finger muscles. Here's another nerve that's leaving the thoracic spinal cord, and it's going to muscles that are holding up the body, the axial muscles. And then here's some nerves that are leaving the lumbar spinal cord and going down to the leg. Now, if we view the spinal cord in terms of these regions, we see that there are 4 regions. There's a cervical region, is a cervical cord, the thoracic region, the lumbar, and the sacral. Sacral, what does that mean? Does anybody know? It means sacred. Most of these names come from medieval Latin, was used to describe things, and so this controls sexual organs, and so that's why it was called sacred. So if you look up here, the cervical spinal cord, the cervical spinal cord has axons that leave the cord. So here's C1 and C2 and C3 nerves, and these nerves are going to the motor nuclei that control your breathing as well as your head and neck movement. So your breathing is done with what? Which muscle? Diaphragm. And so the diaphragm is down here, right? And yet these nerves are leaving at the level of C1 and C2, which is way up here. So why is that? Why are those nerves leaving the spinal cord so soon? Yeah?
I would guess it helps mitigate against injury. So if you injure part of your spinal cord, chances are it's going to be below where those neurons are leaving the spinal cord. So you'll keep breathing.
So that's a good point. That's true. But I think there's another reason for it. So that is true. That indeed most damage that comes to our spinal cord is from car accidents. People get whiplash. And that's at the level of the C5, C6. So when they get paralyzed, they can still breathe. But the reason has more to do with evolution than it does with the idea of protecting the body. And it has to do with the fact that our spinal cord is just along the evolutionary path to other animals. And the very first animals that had a spinal structure were fish. And where did they have their breathing structure? Up here. Yeah, in the neck area, the gills. And so it's one of the examples, we think, of the fact that as evolution took place and animals developed so that they no longer were breathing underwater, but they had this other muscle that made them breathe out of water, that same nerve is now going to the places that controls our breathing. It just happens to be something that's leaving much earlier. Right below it, you see heart rate and shoulder movements, and then wrist and elbow movements. So people that might have a spinal cord injury due to whiplash, they may lose the connection here at the level of C5 or C6. They might become paralyzed, and that would make it so that they have trouble with their hand and finger movements, which are at the level of C7, C8, and the first thoracic level, but they may be okay with the shoulder movements and then wrist and elbow movements. And then you down here have foot motion and other control. So the main message from this slide is that we have the cervical, the thoracic, the lumbar, and the sacral division of the spinal cord, and some idea about the fact that most of the more rostral parts of the spinal cord are controlling your head, your neck, and your arms. And as we get further in the caudal regions of the spinal cord, we get things that control our legs. Okay? All right. Let's take a slice now through the spinal cord. And this is going to give us an idea about the locations where the motor structures are and the sensory structures are in a single layer, where the neurons are as well as the support neurons, the glia neurons are. And the glia are important because they're going to play an important role when we talk about spinal cord damage that takes place in accidents. So this slice to the spinal cord is just a slice that's going like this, and we have the dorsal part, the back, and then we have the ventral part up here. So this butterfly region in the middle here, this is where the neurons, the cell bodies of the neurons are residing. And in the ventral part of the spinal cord is the location where we have the motor neurons. And the dorsal part is a location where the sensory neurons bring in their information. So the ventral horn here, there's a motor neuron here that sends its axon out to the muscle, say the muscle of your finger that causes flexion of the finger. The dorsal root has the cell body out here. Its input is coming from the finger. This is how it's sensing that something touched the finger, gets to the cell body, generates further action potentials that go out and bring that information into the spinal cord. So that information, when it reaches the spinal cord, it might go to the motor neurons, or it might be sent via these axons up to the brain. So outside of this butterfly region, this region that's called the white matter, this is where you have the axons that are traveling from the brain down to the spinal cord, like this blue axons here, or going from the spinal cord up to the brain, the ascending track of the axons. So sensory information going up, motor information coming down to cause movements. And here's the nerve bundle that's leaving the spinal cord to go out to the finger. In addition to the neurons that are sitting here, that are acting on the muscles or receiving information from the muscle, there are these support cells, these glia cells that are astrocytes and oligodendrocytes that provide an important function, which is to produce that myelin that wraps itself around the axons and makes it so that they are insulated, allowing for transmission of the action potentials. Now, let's look at what happens in spinal cord injury. So say there's a car accident and there was a whiplash which caused damage to, say, C5, C6 region of the spinal cord. So here's a picture of the spinal cord. So this is rostral, this is caudal, this is down towards your leg, this is up towards your head. Now the damage is here. This is where the nerve was twisted, causing death of some of the neurons. So now what happens when there's damage like this to the spinal cord? The first thing that happens is that the initial damage area is likely quite small. So the neurons here are going to be damaged, and the blood vessels that are running through here, they're going to break. The blood vessels, when they break, they seep out that blood, and that causes pressure onto the spinal cord. And it's that pressure that then acts on those myelin-generating glia cells and prevents them from receiving oxygen. So this cyst forms, this fluid-filled cavity forms because of the breaking of those blood vessels. And now you have this fluid that has expanded and is putting pressure on these cells that are providing myelin for these axons. So the neurons here died, but because these cells got pressured on them, what happened was that this axon that was running here, which hadn't been damaged, loses its myelin. So when that axon loses its myelin, it no longer can communicate to cells that are below or above. So loss of myelin now causes inability of the brain to talk to these neurons down below, which are just fine, or receive sensory information from the neurons that are down below. So the fluid-filled cavity causes a problem. The second thing that happens is that those neurons that were injured here, they release glutamate. Glutamate is what kind of neurotransmitter is it? Excitatory. It's an excitatory neurotransmitter. There's this flood of glutamate that's released by those neurons, and this causes overexcitation of these neighboring neurons that are out here, which otherwise would be just fine. So this overexcitation causes damage to those cells as well. So the damage initially was restricted to this region, but now you get the fluid that causes damage to the myelin cells, as well as the neurotransmitter release that causes damage to these cells, to the neurons as well. So those two kinds of damage combine to give you a scenario where you have paralysis. You don't just lose the response from these neurons, you also lose the response from the neighboring neurons, as well as the inability to communicate with the neurons that are below and above. So much of what happens in trying to limit the damage associated with spinal cord injury is that to prevent this formation of this fluid, the cyst here, to try to reduce that, to remove some of that pressure, and also to put in antagonist drugs to this glutamate that's being released to prevent this overexcitation of the neighboring neurons. Any questions? Okay, so the rest of the cells are fine. So the cells below, the neurons that are below, are fine. It's just that the brain can't reach them because the highway has been severely restricted now, right? So there are only a few axons that can travel and send action potential down below. So what's done to overcome this from an engineering perspective is to use functional electrical stimulation. So in this scenario, which I will show you, there's a sensor that's placed on some part of the body that the patient can control. So if they've lost their ability to control their finger, like this, what takes place is that one installs a sensor, say, on the shoulder. So here's a sensor on the shoulder. So the patient can move their shoulders fine. It's just that they can't control their fingers because they can't send action potentials down the axons that are going down to the motor neurons that control the finger. So suppose that there's an external controller here that has a sensor here. The sensor senses the motion of the shoulder. It says when the shoulder moves up, stimulate these muscles. So here's the implanted stimulator that receives its control from here that then activates those muscles. And so that makes it so that the muscles can be activated through motion of a shoulder. And let me show you that. Okay. Questions? Yeah.
So, when they're just holding the book, do they still have to like move their shoulders up?
So there are smart sensors that you can put in there and algorithms that you can make it so that they don't have to do it. So they can just do double or basically make it so that they don't have to keep holding this up, so that you relieve some of that extra activity that you would have to have through software, basically. Good question. Any other questions? Yeah.
So there's no afferent information.
Right. Right. That's right. So he can only see what's happening. He's lost both significant amount of sensory feedback as well as motor output. So you see weakness here. So why is it weak? Because the motor cortex is sending all kinds of information, activate, but only a fraction of it is reaching the motor neurons. Yeah.
Is there a risk of motor muscle?
Sure, because the stimulation is external, and if it's too much, then you could damage the muscle. Typically, though, the problem is otherwise. The problem is that the lack of use causes atrophy of the muscles. So this stimulation is useful in reducing some of that lack of use that's generated that causes atrophy. All right, good. Okay, so the muscles were fine. The motor neurons are fine in spinal cord injury. It's just that the communication between the brain and that region has been cut off because of the freeway that are the axons that move from the brain to the spinal cord. They get demyelinated. So in this slide, you have some information about the anatomy of the cord. So here's a slice through the spinal cord, and say this is a cervical region. So you have the ventral part. Ventral is in front, dorsal is in the back. So ventral part of the spinal cord, here we have the ventral part. You have motor neurons that are sitting here sending their axons out to the ipsilateral, the same side muscles. So the left side of the spinal cord sends axons out to the left part of the body, and the right does it to the right as well. So this is called the ventral horn of the spinal cord, and we have motor neurons that reside in the medial part of the cord and the lateral part of the cord. And then out here in the dorsal root ganglion are the cell bodies for the afferents. So the afferents are the things that let you feel something that's touching you, temperature of things that are touching you, the sense of itch that you might have, and so forth. So the cell body for the afferents sits outside the spinal cord in a region called the dorsal root ganglion. And it sends its axons in, so impinge on the motor neurons. Now, as I showed you in the previous slide, there are motor neurons that are in the medial part of the spinal cord and motor neurons in the lateral part. And it turns out that they go to different muscles. The medial muscles go to the medial parts of your body, the proximal muscles, the things that hold up your body. The lateral part has motor neurons that go out to the distal parts, things like your fingers. And easy to remember by this little cartoon. Now, the reason why I show you this is because that also tells you something about the anatomy of the axons that are going up and down the cord. So if there are projections from the brain in this region and then they go out to the motor neurons, these are going to come from areas that are important for controlling your proximal muscles, things that hold up your body, posture. So we're going to see that regions that send axons down in the medial part of the spinal cord are important for control of posture. Whereas things like motor cortex, where it's going to be important for controlling your limbs for movement, like your fingers and so forth, that's going to be the lateral part of the spinal cord, where it's going to make projections out to the motor neurons that are lateral. Questions? Okay. So a single motor neuron goes out and sends axons to a group of muscle fibers. Those muscle fibers always receive input from that single motor neuron. This is called a motor unit. A motor unit consists of a single motor neuron and a bunch of muscle fibers that it controls. So for example, for your gastrocnemius muscle down here, you might have a motor unit that consists of about 10,000 muscle fibers. A single motor neuron going to 10,000 muscle fibers. For the muscle of your eye or muscles of your finger, you might have one motor neuron that goes to 100 muscle fibers. That means that with your finger, you have much finer control of force than you do for a large muscle like the gastrocnemius. So these are the muscle fibers that are innervated by that single motor neuron in the spinal cord. And this is called a motor unit. All right. Now that we know something about a motor unit, I can tell you something about a disease that affects the motor units. This is called polio. Polio is a virus that enters the nervous system through the neuromuscular junction. The neuromuscular junction is here between the axons of the motor neuron and the muscle fiber. So here's a muscle fiber here, and here's the axon of that motor neuron. So the virus enters through the neuromuscular junction, then it travels up to the cell body of the motor neuron in the spinal cord and destroys it. So there you lose the motor neuron, and when you lose the motor neuron, what happens is that you can no longer control all the muscle fibers that were the children of that motor neuron. Because when that motor neuron dies, it can no longer send any action potentials to any of those muscle fibers. So initially, in polio, you have weakness because basically you're losing the ability to recruit those muscle fibers. And what happens is that as the disease progresses, you can't grow new motor neurons, but what you can grow are new axons to the existing motor neurons. So you see that here. So these orphaned muscle fibers get taken over by these neighboring healthy motor neurons. And now what happens is that you have your motor unit, which was consisted of, let's say, 1,000 muscle fibers. Now it has grown to have 5,000 muscle fibers, because it has taken over the neighboring regions. So what then happens is that the ability of the nervous system to control force becomes really bad, because every single motor neuron is sending its axons out to a much larger number of muscle fibers than before. So who's the most famous president that had polio? FDR, that's right. Yeah, he could hardly walk. Despite that, he was elected 4 times to office. Okay, any questions about polio? Very good. So, motor neurons that we've been describing so far are a primary mechanism by which our nervous system controls force in our muscle. It activates those muscles. Well, it turns out that muscles also house sensory organs, and there are 2 kinds of muscle fibers. The ones that produce force are called the extrafusal fibers. These are the things that produce force. They are alpha motor neurons, the large motor neurons that have these motor pools, motor units that generate force. But there are these other kinds of muscle fibers called intrafusal fibers that you see here, that their job is to tell you the length of those extrafusal fibers. So intrafusal fibers are a part of the sensory system. The intrafusal fibers are sitting attached to these extrafusal fibers, and they have this fluid-filled region in the middle, which basically is wrapping itself around by an axon. So when this gets lengthened or shortened, the axon that has wrapped itself around it sends a signal to the spinal cord that says, what is the length of this muscle? So you have the large fibers that generate force, and then you have these small fibers called intrafusal fibers that are part of the sensory system. As the large fiber gets bigger or smaller, so does the small fiber. The small fiber, called intrafusal fibers, have axons that are wrapping themselves around it, and these are called muscle spindles. They tell the nervous system how long is the length of the large fiber. So when my arm is stretched out like this, my spindles that are on my biceps are really stretched out. They're telling my nervous system my arm is extended. When my arm is like this, the same muscle spindles that are on my biceps now is shorter, those same spindles fire less. They tell my nervous system that my muscle is not as long as before, and that's how I have a sensation of where my body is, through those afferents. So it turns out that intrafusal fibers, these things that are controlling the sensory system are also controlled by the motor system. They're controlled by another motor neuron called gamma motor neurons. And in my next lecture, I'm going to tell you much more about the gamma motor neurons and the way they control the sensory system. But for now, what I need you to know is that there are these force-producing muscle fibers called extrafusal muscle fibers that are controlled by alpha motor neurons. And then there are these sensory type of muscle fibers, which are called intrafusal fibers, and they're controlled by gamma motor neurons. There are diseases that affect this. So there's a disease that's called large fiber neuropathy. Large fiber neuropathy causes a death of the sensory afferents. So the neurons that send their axons to these muscle spindles, they die. And so those patients lose the ability to tell where their body is in space. Typically, the disease begins with the most distal things, so like your fingers and so forth, and makes it so that the patient cannot know where their body is in space unless they look at it. They just don't have a sensation. It's like being numb all the time. It's called large fiber neuropathy, a disease that affects sensory neurons. This is in contrast to polio, which affects the motor neurons. All right. So alpha motor neurons, when they discharge, they send an action potential down the nerve that then gets to the muscle fiber that produces a twitch. So here's an action potential in a motor neuron, and here's the force that's generated by the muscle fibers that it's attached to. So you see a little twitch here, and you notice that the action potential has a time scale of about 1 millisecond, but the twitch, which is the force that's generated by the muscle fiber, has a time scale of about 30, 40, or 100 milliseconds. So the force that's generated is much slower than the single action potential. So when the action potentials come close to each other, so in this case you have, they're separated by 5 milliseconds, it's a 20 hertz activity, what you see is that you get this buildup of activity. Sorry, 50 milliseconds, you see this buildup of activity. At 80 hertz, it has less time for the force to decay, it gets built up some more, and then when the motor neuron is firing maximally, say at 100 hertz, now the force has no opportunity to decay, the twitch never falls, and so you get this condition where it's called tetanus. That's when you're maximally activating. So one way by which your nervous system can control the force that's being generated is by rate of activity in the motor neurons. When the rate is higher, there's greater amount of force that's generated. All right. Now, another way by which the nervous system controls force is the length of the muscle. So here's the fundamental difference between biology and engineering. So when you go and you buy a motor to move something, a little robotic arm or something, you generate a voltage, you give a voltage to the motor, and it will generate a torque for you. And it doesn't matter where the location of the rotor is, it will always give you the same torque. Muscles don't behave that way. If you give 100 hertz stimuli to a muscle, it will produce a force, but the force that it will produce depends on its length. The longer the length of the muscle is, the greater force it will produce for that same input. This example is shown for the soleus muscle, which is the muscle on your ankle down here, that shows you how the input that's generated, the stimulation of that muscle, will produce a greater amount of force depending on the length of the muscle. So in this curve, what you have is that the muscle is maximally stimulated. So it's at tetanus, say 100 hertz. When that maximum stimulation is given and the muscle length is short, it only produces about a quarter of its maximum response. So here's about a quarter of a kilogram, a half of a kilogram. But when that same 100 hertz is given, when the muscle length is long, it produces much greater force. So the force that's generated depends on two things. How many spikes the motor neuron is generating, the rate of activity, and the second, what's the length of the muscle. So the control of force in the muscles needs to know the length of the muscle. You need to know what is the length of your muscles before you can generate force. That information about length comes to the spinal cord via those spindle afferents. Any questions? Yeah.
Talking
So good question. Thank you for bringing that up. So the ankle here has an angle which is shown here. So as this angle becomes smaller, your foot is being raised, which means that the soleus muscle is getting stretched. Yeah. Right. So beyond a certain length, it begins to fall. So there's a maximum, there's an optimum location for it to generate a force, and then a little bit longer than that, it falls. And it has to do with the sarcomeres. As they get too far apart, they have less to this optimal location, and then that's where the maximum force begins to drop. Good question. Any other questions? Excellent. All right. So in this slide and next, I want to reiterate what we learned about how to go from force generated in the muscles to torques generated in the joints. So I have a little cartoon here of your elbow. Here's a muscle. This is like your triceps muscle, and it's going to act on your elbow to pull on it to cause a torque, which would cause extension. And what we want to know is that how we're going to relate the force that's generated in this muscle to the torques that are going to be felt. And the relationship has to do with that term that I described for you a few lectures ago called the Jacobian. The Jacobian is the relationship between the length in the muscle and the position that you're looking at. So for in this case, the joint angle. So what we want to know is that what's the relationship between force and torque. So what the Jacobian tells us is to look for the relationship between length in this coordinate system and length in this coordinate system, which is the joint angle. Okay? So to go from force to torque, what I need to do is to go from position in this coordinate system, which is the length of this muscle, to position in this coordinate system, which is the angle of this joint. And that differential transformation is called the Jacobian. d lambda d theta. Change in this length as a function of change in this angle. So to do that, what we need to do is write the muscle length as a function of the joint angle, which is done here, using law of cosines. So the length of this muscle as a function of this angle is shown here. And d lambda d theta is the derivative of that length with respect to theta. So the derivative of the square root function. So how do I find the derivative of that? Well, I find the derivative of anything that's raised to 1/2, and then times the derivative of what's inside, which is shown here. So the derivative of something raised to the 1/2 is just that thing raised to the -1/2, so that's why you see this in the denominator, times the derivative of what's inside with respect to theta, which is this term cosine theta, which becomes sine theta. So this is the Jacobian. This tells us how we can go from force generated in this coordinates to torque generated in that coordinate. So I take force and multiply it by this to get the torque. Yes, sir?
Where's the 2? Shouldn't there be a 2 in the numerator there?
Yeah, it's going to get, because there's 2 here, it's going to get canceled.
Oh, right.
Good question. Good. Okay. So this is the same as a moment arm, right? This is just a fancy way of writing what a moment arm is. And basically, that's this length, this perpendicular to this length here. If you plot d lambda d theta as a function of theta, you see that it has a maximum value at 90 degrees, which is the angle that gives you the maximum transformation from force to torque. And just to show you that this is indeed the moment arm, in the next slide what I've done is that I've just recomputed this for this length here, c, which is what you would normally do to find a moment arm. And you see that this is exactly the same as what we just did in the previous slide. That's also d lambda d theta. So I'll leave you to take a look at this slide on your own, just to convince yourself that this length is indeed the derivative of this length with respect to this angle. All right, that's called the Jacobian. So what we have in our body are antagonist structures of muscle. So you have biceps that will pull on your elbow, and you have triceps that will pull on your elbow. So you have a force here that's going to pull, and you have a force here that's going to pull. So what you do to compute the torque that's going to be generated by these two muscles, where you take the torque that's generated by this muscle, that's called tau 2, and the torque that's generated by this muscle, called tau 1, and you add those two and you get the torque that's generated by the sum of the forces generated by each muscle. So if I have force F1 and I have force F2, by multiplying it by these Jacobians, I can compute their torques, and then I add them and I get the sum total. Okay? And so your maintenance of posture, your ability to hold still at a particular location, comes about because there's a torque that's generated by this muscle and there's a torque that's generated by this muscle. The two happen to precisely cancel each other. And there you get steady holding. Now, you might think that holding still is a simple task because, you know, movement you might think is difficult. But it turns out that in your nervous system, there are specialized circuits that allow you to hold still, making it so that you don't have tremor, which comes about when there's damage to the circuitry that is important for balancing out the agonist and the antagonist precisely so that the arm stays still. Because if you imagine, if your agonist is activated a little bit more, you might start drifting up, and then the antagonist would get activated, it would go down, and that's how you get tremor. Only when they're perfectly balanced, you get this rather remarkable ability to hold perfectly still. And otherwise, you know, you can't do it, which is another indication of really how complicated our nervous system has to work to maintain posture. So posture comes about because of this perfect cancellation of the forces that are generated by the two muscles. So you can move between two postures by just changing the activity of these two muscles. So at this location, activity of the biceps and triceps can get canceled to produce this zero force. Now, in order for me to move here, what I need to do is activate the biceps so I get to this location and hold at that location. So as you can see it in this example, say that there's input to these two muscles, U1 and U2, both of them say at 15 hertz, and the muscles are of equal strength. What will happen then is that the arm will come to an equilibrium no matter where it starts. So if the arm is down here or the arm is down here, in either case, you will get to this equilibrium that is represented by the forces that are generated by these two muscles. It's like two springs. These two springs will generate identical forces, and so they cancel each other, but only at one location. And so if the arm starts here, it'll move to here. If the arm starts here, it'll still move to there, because that's the location where the torques will cancel. That's what gives you this posture. Regardless of the starting position, as a constant input to the muscles, the limb converges to a single equilibrium position. So because you have muscles, it makes it so that when something hits your arm, it naturally will resist it. Why? Because if something pulls on this arm, pushes it downward, this muscle here will get stretched. And because it gets stretched, it produces greater force. So it naturally will pull up. Even if you have no sensory feedback, just because you have muscles, the mechanical properties of the muscles gives you this equilibrium. You have stability at that location. Any questions? Yeah.
So is there always going to be greater force produced when our muscle is lengthened? Like, it's easier for me to hold my arm out like this than I feel like it is for me to hold my arm like this.
Easier in the sense of?
Or I guess if I'm standing up, it's easier for me to just
let it dangle, right?
For me to hold that.
Right. So your sense of effort, we think it's coming from the activity in those neurons. So the sum of activity in those two neurons probably is least when your arm is here, and probably the sum of activity in all the neurons is more here, because there's something that's holding it up against gravity. Great. Okay. So how do you make movements? Well, the way we move our arm is through a sequence of activity in the muscles. To make a movement from, say, here to here, where my elbow is flexing, what I need to do is activate the biceps to begin accelerating the arm, and then to stop the motion, I need to activate the triceps to stop it, and then there's always a little external activity of the biceps to hold it at that final position. So it's an agonist. It looks like this: agonist, antagonist, and then agonist. Agonist, antagonist, and then a little bit of activity in the agonist. So to make a rapid movement, biceps, triceps, and then back to biceps for the small activity. It's like when you are at the red light. To rapidly move, you accelerate, then you put on the brakes, and then a nudge at the end to get to the next red light. It's called the 3-burst activity: biceps, triceps, back to a biceps. This is how you make fast movements. So that was my little cartoon of it. Let me show it to you with actual data. In this example, what we have is a wrist that's making a rapid movement. So here's position of the wrist, here's the velocity of the wrist, and here's the activity in the muscles. So here's the wrist agonist. This is the burst here. And then here's the antagonist down here. And then finally the agonist again. Agonist, antagonist, agonist. So these bursts of activity in the muscles have to be exquisitely timed. Otherwise, you will not stop at the target, you will overshoot or undershoot the target, or you might have tremor. So that's my final slide to show you that today. Here's an example of a deficit in the cerebellum. So this ability to make rapid movement and stop on the dime on the target is coming because something is helping the motor cortex time the activity of these muscles. So we see an example of it in a condition called essential tremor, which has to do with damage to the cerebellum. So here's a healthy person making a wrist movement, and let's look at what happens. So here's a wrist that they're making a rapid movement. Is the position. Here's the velocity. Here's the agonist, here's the antagonist, and here's the agonist again. So you see that the last burst is coming right around the time when the velocity is going to 0. Now here's a patient that is going to make the same movement, and what happens to them is that they make a rapid movement, but they can't stop. They have this tremor at the end of it. You see it? Here's the velocity that can't stop. It's as if the system is underdamped. They make a movement, but they can't stop it, and there's this velocity, this tremor, this oscillation at the end of it. And it's coming about not because they can't start the movement properly. So they have an agonist that looks reasonable. They have an antagonist that more or less looks reasonable as well, but you see that that final agonist is coming too late. So now it's coming when the velocity is positive rather than back here where it should have been about 50 milliseconds earlier. And this lack of coordination, lack of timing between the muscles causes so that these bursts are coming later and the system isn't stopping. It's oscillating. It's tough making rapid movements. It's really an astonishing, beautiful thing that, throughout our lifetime, we, by and large, unless we have a deficit in a structure like the cerebellum, we're going to make accurate movements for, decades in our life. But just a 50 millisecond shift in when one of the muscles is going to come on is going to cause oscillation. Any questions? Thank you so much. I'll see you Wednesday.